Just want to check if my mic is on. It is? Awesome. I see nods. So before we start, I guess we meet here today on the lands of the Wurundjeri people of the Kulin Nation, and we pay respect to their elders, past, present, and emerging, and elders that might be here with us today. Thanks everyone for coming. It's probably dinner time, and you probably want to go home to where there's more food and more warmth. But thanks everyone for being here. Hopefully, we can give you some insights into some of the activities through the University of Melbourne. My name is Christina. I'm here representing Illumina. Some of you might know who Illumina are, and if you don't, you'll probably know a bit more by the end of today. I guess a brief introduction on what today's going to be. I'll just take you through a little bit of a background between the partnership, which you'll know is either called TAGC or the Advanced Genomics Collaboration, and we'll use those words interchangeably tonight. And that partnership is between Illumina and the University of Melbourne, and it's supported for innovation projects, which is going to be the topic tonight. And the three people here, which I'll introduce, will be talking pretty in depth about their innovation project, either what they're working on today and possibly a bit on some of their other genomics projects as well, not limited to just sort of what you're doing. And so just to sort of kick it off, we'll have a panel later as well. There'll be a lot of questions that I will ask. They'll start very high level and get very granular, but I guess we'd want you to ask some questions at the end as well. I think there are a few Illumina interns in the room possibly, so I'd probably expect you guys to ask some questions as well. No pressure. I think you guys should be first. So a brief introduction to Illumina for those who may not be familiar with us. We are a global genomics company. We are founded in 1998, which was, I guess, the start of the Human Genome Project as well. We're based in San Diego as our HQ. We have about just over 9,000 employees. We focus fundamentally on genomics and very specifically next generation sequencing as well as genotyping. We have major R&D hubs in San Diego and Cambridge in the UK as well as in Singapore. And we play a key role in, I guess, precision medicine, which is a bit of a buzzword these days. And we serve lots of different markets. So we serve, in terms of precision medicine, we serve the oncology markets, the rare disease market, but also other markets such as microbiology, infectious disease, and also agriculture. So genomics sort of spans every organism you can think of, including the atmosphere and waterways and things like that. So there's so much genomics activity, but I guess a lot of it is probably not, hopefully you learn a bit more today, that's something different. In terms of our customers that we serve, we do a lot of work with universities and hospitals, as well as pharmaceutical companies, service providers, as well as entrepreneurs and startup companies. Our mission is to unlock the power of the genome, and a lot of people who work at Illumina, if you've had a chance to meet with them, really do resonate with that mission. But we can't do it unless we partner with others to help us execute on that. And so I guess that's a good segue into our partnership with the University of Melbourne, which is called the Advanced Genomics Collaboration, or TAGC. It was founded in 2019. And the purpose of that was to really sort of support genomics from a university or a translational level, but to really push it towards a clinical outcomes and that precision medicine. It was supported through the Victorian government, specifically Invest Victoria and the Victorian Department of Education and Training. And the partnership is really to offer affordable, scalable, accurate, and I guess innovative technology in terms of genomics. And you'll hear about a lot of those techniques today from our panelists. I guess together we're working to really accelerate biomedical research, making genomics sort of accessible, and the end goal is to really improve human health for all. And so the TAGC is underpinned, at the start of the partnership, was to set up three core enabling platforms. The first one was the clinical genomics platform, which is essentially a sequencing platform to deliver rapid whole genome sequencing. It is at a global scale and a global quality as well. The second platform was an informatics platform. It delivers a very rapid, affordable, scalable creation of data storage and also data interrogation as well. Again, all that extreme scale, extreme speed, and extreme efficiency. So if you don't know too much about those platforms, I definitely encourage you to either speak to some of the folks here today, or there's even a website that has a lot of information as well, which you'll see on the screen later. The third platform is a health economics platform, and I guess it really wraps the two earlier platforms into that evidence generation, which then starts to infer sort of policy and a lot of that, which then moves into sort of making real change into the community. So with that, those three platforms, then now we have these innovation programs, which they then utilise either one or all three of those platforms in terms of the sequencing, the informatics, or the health economics piece. Some of them use all three, and you'll hear a lot about that today. So we'll be discussing today, as I said, next generation sequencing predominantly, how it's really revolutionising research and patient care, and really ushering into that era of precision medicine, which you'll probably hear repeated many times, and that's always a good thing. So the 4 TAGC innovation projects we have, you'll notice one panel member missing today. Unfortunately, they do send their apology. But the 3 that you'll see here, the 4 of them collectively are awarded, or sharing at least, $6.5 million in funding over 2 years. They address either cancer, inherited disease, or common gynaecological disorders. So before we start the panel, we'll play a very short video. It's only a couple of minutes. It'll give you a background into the 4 different projects, and it'll hopefully give you a bit of insight before we start asking some questions. So thank you.
Illumina and the University of Melbourne are working together to unlock the power of the genome and reimagine what's possible for clinicians by transforming how they detect, diagnose and treat diseases like cancer. With the support of Invest Victoria and a diverse network of skilled collaborators, we are building a pipeline of genomics-focused innovation projects that can make a step change to patient care.
We receive support from the TAGC Innovation Fund to pioneer a less invasive way to collect tumour information from a simple blood test. The goal of this test is to detect and decipher the small amounts of cancer-specific mutations found in a patient's bloodstream.
Our research is really around trying to identify people with inherited retinal diseases and learning more about their clinical presentation as well as their genes. Until quite recently, there were no treatments at all for inherited retinal diseases, so all we could do would be offer people things like long canes or guide dogs and extra mobility training, whereas now we actually are at the precipice of a whole raft of new treatments. With the TAGC Innovation Project, we're now going to be able to learn more about these people, what the genetic differences are in their bodies that cause their eye disease, hopefully target new treatment options, and also identify people that could go into clinical trials now.
Cancer of a non-primary is an important clinical problem. It is an uncommon condition, but it affects 2,700 patients every year in Australia alone. Knowing what the cancer type a patient may have is really important to directing the best treatment for that patient. But it also causes increased psychological distress for the patient, not knowing where their cancer arose from. So through the TAGC Innovation Project, we're developing a new blood-based test that can potentially solve that diagnostic dilemma, as well as direct a new treatment for these patients. The advantages of a minimally invasive blood test is the speed of testing, but it also allows testing for patients who might not otherwise be able to access that form of test. This project would not be possible without dedicated funding from the TAGC, both to support the platforms themselves and also the team of people that work on this project.
So put simply, our TAGC Innovation Project is a tissue banking project. We're going to recruit women, we're going to collect bloods and tissues from them, which we're going to do genomics on. That allows us to give more appropriate and targeted treatments to individual women. We already had funded research in endometriosis, and we were recruiting patients into that project, but this has allowed us to more than double our recruitment rates and to include other ailments such as heavy menstrual bleeding and recurrent implantation failure into our database. It's generally recognised that there are opportunities in women's health that are unmet, and that there's certainly an opportunity to try and do something about that.
Genomics is transforming healthcare because it's revolutionising our ability to be able to understand the causes of different diseases. So for the inherited retinal diseases that we work with, it helps give information about family planning, but also gives those affected the ability to understand what is causing their condition, and that knowledge is powerful.
Liquid biopsies have the potential to offer cutting-edge genomic testing and treatment options to patients who do not have tissue available, allowing us to provide diagnostic certainty and actionable insights for thousands of additional cancer patients who might otherwise fall through the cracks every year.
It's a pretty amazing feeling. It is quite amazing to see how quickly this field has changed from where we were 10 or 15 years ago. The fact that we can now look at somebody's whole genetic information and help that explain what they're experiencing as a disease is just mind-boggling.
This collaboration is a great example of the magic that happens when some of the world's best organisations and minds come together to solve some of the world's largest problems.
Shout out to the marketing team on that video. Thank you, Zoe. So just, I've sort of introduced them, but officially in 3D, we have Professor Lauren Ayton from the Centre of Eye Research Australia and the University of Melbourne Department of Optometry and Vision Sciences. In the middle, we have Associate Professor Richard Tothill from the University of Melbourne Centre for Cancer Research and the Department of Clinical Pathology, and then Professor Peter Rogers from the Women's and Children's Hospital and the University Department of Obstetrics and Gynaecology. So I'm going to pick on everyone one at a time, but you can always jump in as well. Lauren, I'll start with you, ladies first. Your project is very much looking at inherited retinal eye disease, and you mentioned in your video, classically you would use guide dogs and other non-genomics-based sort of techniques. Now this project is using genomics, and how has that changed your team that you now work with, and can you tell us how it sort of aims to change routine care? We're going to get into a lot of detail, so you can feel free to keep it brief, and then we can unpack some of those challenges and things later.
Yeah, absolutely. So as I mentioned in the video, so inherited retinal diseases are the most common cause of blindness in working-aged people. So everyone in the audience, if you were blind, it would most likely be from one of these inherited diseases. The challenge is there's over 300 different genes that can cause an IRD, and up until quite recently, we didn't do genetic testing. It wasn't a typical clinical tool, and so there were no treatments for IRDs, and there was really not much hope we could offer patients. There are now gene therapies, and really excitingly, the first ever direct gene therapy for a patient is for an eye disease, one of these inherited retinal diseases. And so we now have drugs on the market, more coming up in clinical trials, but we know that less than 10% of people with these conditions in Australia know their genetic information. And so this is really what drove this particular innovation project, to try and answer the other 90% of cases.
We're going to tap into some of the unsolved cases later, so thank you. Richard, I'll go in order. You're sort of the liquid biopsy guy. You could call yourself that if you feel like it, or maybe I can label you that. Similar to Lauren, I guess, can you just give us a brief intro on your project and sort of the team that underpins that project as well?
Yeah, for sure. So as I mentioned in the video, I work on using genomics for cancer diagnosis. So in most cases, patients will get an accurate diagnosis. But in a small percentage of patients, we don't know where the cancer arose in a patient, and really knowing where the cancer came from is important for directing the best treatment, but also can impact access to treatments as well, as most treatments are funded by the federal government for specific cancer types. So cancer of a non-primary, where we can't resolve the cancer primary site, does create a bit of a dilemma for clinicians, and also causes a lot of psychological impact for the patients as well. So we've been using genomics for some time in helping to resolve the cancer of non-primary and identify what the primary site is, and more recently, I guess, looking for precision targets, looking for particular mutations in a cancer that will direct a specific treatment. So these precision treatments are becoming more common, and we need to do DNA sequencing of a patient's cancer to identify that. So we're really using genomics for two purposes: diagnosis and directing treatment, so getting patients onto the best treatment they can. This works really well, although it's still not, this genomic testing for CUP patients is still not, rebated by the government. And I think we're very close, I think, to making that standard. But in many cases, we don't have enough tissue to do the test, or getting the tissue out of archives is really challenging. So this is where liquid biopsy, looking for small amounts of DNA that's released from tumour cells into the patient blood, can be really helpful, and can be a substitute for looking at tissue. It can be much quicker as well. And so we're hoping that using a liquid biopsy test will facilitate more rapid testing for these patients.
You used a word, CUP patients. For people in the audience that don't know what a CUP patient is, maybe you can explain some of the ideology behind that?
Sure, sorry. Well, cancer of unknown primary. CUP's just an acronym for cancer of unknown primary, of course. So if I do use the word CUP or the acronym, just know that that's cancer of unknown primary.
Another thing you said was, I guess, the psychology of knowing a diagnosis, and I think that's probably relevant for all of the projects here. It's something that we talk about a lot at Illumina as well, is even though you might not have a treatment associated with your diagnosis, just understanding what your diagnosis is really stops that testing paradigm. Some people go through, 7 years worth of diagnosis to find out what it is that they actually have. Any one of you who have a family member or personally who you just want to know what is the cause of X, Y, Z, sometimes knowing, aside from a treatment, is also good as well. Peter, same question to you, a bit of a background on, I guess, the project and I guess the team that is behind all of that as well, if you could, please.
Thanks, Christina. So we've, at the Women's, we've been working on endometriosis since I came to the Women's in 2010, and that had a significant genomic component, very significant. So the endometrium, or the lining of the uterus, there's bound to be at least one person in the room, possibly male, that doesn't know what the endometrium is. I always use the word volatile, and I mean that in a sort of a slightly amazed way. The lining of the uterus changes so dramatically during the cycle that it's almost impossible to study, because you don't know where it's at when you get a sample. It's changed, and it changes by the hour. And to make it more confusing, the cycle length varies dramatically between women, and even in the same woman from cycle to cycle. So this is a really difficult thing to study. Now, it turns out that the genomic profile is actually very consistent, and you can use it if you stop worrying about what day of the cycle it is and look at the genomic signature. You can get a very accurate feel for where the endometrium is. Once you can do that, you can take the endometrium and compare it between a woman who has a problem, for example, infertility or endometriosis or heavy menstrual bleeding, with a woman who doesn't have a problem, and you can normalize the gene expression so that you can actually start to make valid comparisons. So this has opened the window to a whole lot of potential precision medicine, diagnostics and therapeutics, because we've now, I say tamed the endometrium, but we've at least got a handle on what it's doing, and a very precise. There's thousands of genes that change significantly on a day-by-day and hour-by-hour basis during the cycle as the endometrium repairs from menstruation, then grows under the influence of estrogen, then differentiates under progesterone in expectation of embryo implantation before cruising for a little while and then suddenly preparing to menstruate again. And these changes are literally daily. So genomics has had a massive influence there. That work, we published that last year, but the university patented it before it was published because it was novel enough. And I guess we're now building on that, and we have commercial interests in it as well as, what the TAGC funding has enabled us to do is, just as it said in the video, start expanding to other, including endometriosis, but expanding to look at other conditions that basically affect nearly every woman at some stage during her life. So it's very broadly applicable.
Thank you, everyone. I might direct this question at you, Lauren. Only because I know that you engage with, I guess, the community and, I guess, health consumers. How is that sort of incorporate into this project? Is it different in any way, or is it sort of similar?
Yeah, I actually was just reflecting when you said before, Christina, about, you know, the power and the empowerment, I think, of having a diagnosis. And that is a really key thing for people with these inherited retinal diseases. They have an answer for what has driven them. And so this was actually a big part of why we've actually gone down this route. So my team historically were very clinical in our research. So we actually didn't have genomics people within our team. And then over time, we started to bring that expertise in because we were getting reports from patients that it's really important to them that they know their diagnosis. And for us as clinicians, it's really important that we can identify which patients are most eligible for treatments. And so we're building up a big database of people that are suitable for these gene therapies as they come up. And so we obviously wanted to have that genomic information. But yeah, everything we do is grounded by our community that support us, and we're, you know, involved in different committees, and we have consumer advocates involved in all of our projects. It's, yeah, very important to us.
Richard, this one's for you. So every time I talk to Richard, he's either written a grant, writing a grant, or just submitted a grant. And I know there's a lot of researchers in the room. So
the academic's life.
So I thought maybe you could give, in terms of genomics, I know you write a lot of genomics-focused grants or multi-omics grants. How is the innovation project and this particular grant different from, I guess, the others? And, you know, how was that sort of... maybe is it more work or less because it's led you here today? So maybe you could give us a bit of a synopsis on the TAGC sort of innovation grant specifically.
Yeah, I think so. When you write a grant, you have to tailor that to the particular scheme, and so not all grant schemes would be appropriate for the project that we put forward for the innovation study. So I guess the idea here is developing a commercial test. And it's very much a translational. It's not very much a discovery, although I think we're going to make discoveries along the way because we're starting to really do genomics at scale. And you don't know what you'll find. There's a lot of serendipity involved with that. But if your goal is to develop a diagnostic test, not all schemes would be appropriate for that. So I think it's been really important. And it's clearly, you know, we're doing things at scale, involves... I didn't talk about the team, and I should have. There's a number of people working on this study across 2 laboratories. So my laboratory, which is the Rare Disease Oncogenomics in the centre, and Sarah Jane Dawson, who's principally at Peter Mac, but now co-located with us in the same cluster area. And so across our 2 teams, we've got 7 different people working in the wet lab side and bioinformatics. And we've also got external collaborators, a not-for-profit organisation called the Hartwig Medical Foundation, and they've been very good collaborators in terms of developing new methods for analysis of the data. So there's lots of people involved with this study, and I think you know, the funding has been incredibly important. It wouldn't have been possible to get this off the ground without the TAGC.
I might just add to that as well. I think we were just talking about this before. One of the huge strengths with this program is the strong interaction that we get to have with Illumina and with the TAGC team. And, you know, we really, I think, get so much more involvement from a standard grant where you kind of get the money and you do the work. You know, this is more than that. It's a partnership, and it's really grown our research space so much.
And I guess maybe touching on that, one of the other things that it involves is also, as a really key part of it, is students and student engagement. And all 3 of you have, I guess, been assigned some students, and there were, again, that was its own selection process. Some of you might be familiar with it. Different again to any other internship or any other sort of university student engagement. Maybe you can tell us, maybe Peter, I'll pick on you first, that the other, you know, feel free to jump in, a bit about, I guess, the student piece and they only just started, right, and it's in the early phase, but maybe you can give a bit of an update on how they're going and what you plan on putting them through for the next maybe 2 years.
So I think it would be really exciting. I try and think back to when I was a student, but it was probably so long ago I lost in the mists of time. But the students have really been given quite a big challenge to understand, I think, the whole process of discovery research through to commercialisation and then implementation, what we call the translation stages from 1 through to 5, which is acceptance by the community of the treatment. And I think that could be a bit of an eye-opener. I mean, you know, you sort of might think of one stage or the other, but how they all work together. And we were talking the other day with them about the role that the researchers sometimes have to play working with the consumers and with government to get the consumer's message to government, so that government then provides the resources. So the advocacy that occurred in endometriosis involved the consumer groups in each state collaring and two MPs on different sides of the House, collaring the health minister, getting Canberra interested. There was a public apology in Parliament, actually. That was Greg Hunt, the minister there. And then there was a national workshop which produced a national statement, which we managed to make sure we got a research component into it, along with an education and a clinical component, and then a round of MRFF funding. So you got to see the whole cycle and understand that actually consumers are empowered to do things, but you need to actually do them, and that requires energy and I guess leadership. The two female MPs, one Liberal, one Labor, provided important political leadership, and I guess having experts always, the next level of political unrest was quashed by the professional people who showed them what the right way was. But these sorts of understanding of the global thing, you can spend a long time in your career and wait for experience to discover those things and to actually learn about them. They're outside, I think, the scope of a lot of your typical university type courses. So I think it's a really good scheme.
Students, yeah, I've been so impressed with these students, the quality of all the applicants actually who applied to be interns. So we've taken on a number of interns, and there's, I guess, a number that we need to take on for the first 6 months and the second 6 months. So we tentatively took on 2 to begin with, to try and feel our way through that. But again, the quality's just, they're just outstanding people. They're doing okay. I guess it's a lot for them to take in. They're coming from, an undergraduate, essentially, one's doing their masters now and the other's in their 3rd year, throwing some challenges at them and seeing how they cope. And they've been taking in their stride. They presented today they discussed their projects, I guess, and what they're going to do, with relatively little input from myself. But it's been fantastic having them. Yeah.
And I think one of the other huge strengths is it's a whole other set of eyes, right? on our projects, because we get so enveloped in what we're doing, and we can become quite short-sighted. I've got lots of eye puns, don't I? It's very much having them come in and say, well, have you thought about this? And we've really been tasking our students with thinking about the future for our projects. If we can come up with some better genomic pipelines for diagnosing these eye diseases, what does that look like? Can we set up a clinic in Victoria? How would we actually implement this? And so they've really been getting their teeth into the future, which is exciting.
Awesome. All right, let's get into some of the granular stuff now. So with, theme of the Innovation Week, the theme of, I guess, technology that is, I guess, developed from industry partners such as Illumina or otherwise, they tend to, I guess, innovate in this technology space. We put these products out there, these instruments out there, and we go, right, what are you going to do with it? So, and then sort of it's the research that comes out of it, the translation, and then that goes into healthcare. But I guess for your particular projects, all of it, they all use next generation sequencing. How is that sort of different to what was done maybe, a decade ago, 2 decades ago, and in terms of 4 decades ago. Look, you brought up the age thing, not me. But what sort of made that project possible today in terms of insights and things compared to... I'm looking at you, I think you should go first.
Yeah, very happy to. So look, I think for us in the ophthalmology space, we weren't doing this 4 decades ago. We've really only started doing genetic testing relatively recently. There's a really amazing graph that I always put in my lectures where the first gene for an inherited retinal disease was discovered in 1981, and it's just gone like that, to have 320 plus genes identified now. So it is a really recent thing for us. it has been really hard for patients to access, because one of our big challenges is a lot of these eye diseases look the same. So if we were doing single gene testing, I mean, it's too expensive to do 300 individual genes. So the fact that we can access panel testing and then we can do whole genome sequencing now is game changing. And one of the things I've been loving with this particular project with Illumina is we're just learning so many new techniques and things that are coming up and haven't even hit Australia yet. We're sort of getting an inside look into those and thinking about what that means for our patients. So it's been quite exciting.
So technology, I've been in this field for more than 20 years now. I've actually worked on CUP, cancer of a non-primary, for more than 20 years as well. And I've seen this evolution of different platforms, from the very early genomics platforms, really the first genomics platforms, right through to where we are now, where we're doing whole genomes for well under $1,000. The technology keeps advancing, it's getting cheaper, and that's really the scalability. We're going broader, deeper, and we're scaling. We're going in higher resolution with some of the single cell technologies, spatial genomics, etc. now as well. So it's really been an amazing journey over that time to see that evolve, and I got to apply these methods in the same kind of context and see where these new technologies can improve things. I think there's been evolution in the molecular side, or the molecular biology and this instrumentation. We've seen a huge advance in computational biology and high performance computing. That's been really essential. And then just the generation of data. So doing what we do now, if we did a whole genome on a patient's cancer 20 years ago, we wouldn't even know what we're looking at, because many of the genes and mutations in the genes, we go, oh, that's interesting. It's a gene called BRAF. What does that do? And now we know that's one of the primary drivers of melanoma and colorectal cancer and other diseases. So we need that data resource and that knowledge base really to apply the technology properly now. And we're starting to use genome-wide sequencing to then look at profiles, not looking at individual genes. We're really looking at profiles of cancer, molecular portraits, and using AI or machine learning to classify cancers and to determine what is driving that cancer, what is the best treatment for that patient.
I think you mentioned something there which sparked a memory or a deep thought in the back of my brain, which is some of your projects as well use, I guess, multi-omics, and that might be a new word for some people here, but it's looking at multiple different samples just for the same patient. So, you've spoken about liquid biopsy, but then there's also the tissue side of things, and using that in multiple different ways, which I think is pretty cool, something that probably no one did many years ago. With that, though, I guess comes more information, and information is power, but I guess, and we'll come to your technology update as well. Maybe I'll stop there before I ask my second question, because I'll let you finish first.
I can run with what you've just said. Multi-omics, we're doing it. Absolutely. I guess the one advantage of being around a very long time is you watch each new technological advance and you listen to all the people singing its praises and how it's all going to be solved now. And this will fix everything. And of course it doesn't, because there's another level of complexity, and each time you unravel that, there's another level of complexity. So now we're talking about multi-omics with AI, and we've actually got investors interested in that. We've already got a proteomics partner. We're doing genomics. We've got the genetics on most of these women from a collaboration. And we've also got some pilot metabolomics work going on. And the data is overwhelming. Who knows if AI will make head or tail of it. But it's where everything's going at high speed. There are some success stories. I'm sure you only hear the success stories normally, so I'm sure there's quite a few disasters out there. But if you don't look, you don't find.
I guess with that, keeping with you, Peter, is what would you see then the barriers to implementation? Like you said, there's all this information, there's all of this new data. From a research perspective, it's cool, it's exciting, but then how do you actually adopt that into routine healthcare? Funding aside, I think what are some of the other sort of...
Absolutely, you've got to have your eye on the whole pathway. It's a bit like taking a car apart down to its constituents nuts and bolts. It doesn't actually help you. You've got to be able to put it back together and get it working again. I think that what the hope of all researchers are that you'll discover the gene or the gene pathway, and then you'll be able to go to the next step and either identify a bit of it that you can use as a diagnostic, preferably easily accessible, so bloodborne, or a therapeutic, so a step in the pathway, preferably very specific with no side effects, and then develop the necessary interventions to deal with it. Just knowing how something works doesn't help you if you can't do anything about it.
Adding to that as well, I think one of the other big challenges with the amount of information that we can get now is what happens if you discover something that you didn't know or you didn't want to know. And this is an issue for us with eye diseases, because quite often the eye diseases are syndromic, and so people might find out, oh, actually you're also going to lose your hearing, or you could have a heart disease, or you could have a brain condition. And so that's a very big clinical issue, because as you say, Peter, you need to have the pipeline in place to keep caring for the people after they get the diagnosis. You need to be able to refer them off. You've got to have counselling, and that's a challenge I think that our system has.
We'll press on, because I think we can probably talk about the ethics part is an interesting one too, but we won't touch on that today. I guess in terms of innovations, we've got that theme. What innovations do you want to see, thinking very broadly, the world is your oyster? What needs to happen ultimately for your project or area of interest to be really successful? It's a big question. Go on, Richard, let's go first.
Look, I mean, there is the cost of sequencing is falling, and I think that will allow us to do more comprehensive testing on patients. I mean, most testing at the moment is more panel-based, I guess, but we're at that cusp now where whole genome is perhaps as cheap in some contexts, especially for sequencing germline, so looking for hereditary genetic traits. But that for our project, we're going deep on liquid biopsy samples where we're looking for very small amounts of DNA in the patient's blood. We need to go deep to find what we're looking for. So I think that'll make things more affordable. I think other than that, for us it's more about we need to validate, we need to show clinical utility. So that's the next big challenge for us. I'm confident that we'll be able to generate a test that works, but it's then showing that it works in the clinic and it actually helps patients. So that's the big challenge for us.
We're very excited within this TAGC project to sort of look at identifying new genes, solving the cases that haven't been solved, those sort of things. But I think what I'm really excited about is that next step of how do we turn that into treatments for these patients. So I mentioned before that there is one gene therapy available for an inherited retinal disease, and that's a gene augmentation therapy. So it only treats patients that have, a biallelic gene mutation on a specific gene, and it's less than 1% of patients that have these diseases. So gene editing, other types of treatments for these eye diseases are coming, and that's really exciting space for us to all be in as well. So all of these treatments need the genetic information. So we have to do the testing first, but then what do we do to treat that patient?
The field's wide open. If you look at the women that walk through the door of the women's hospital every day, better treatments for heavy menstrual bleeding, non-steroidal treatments for heavy menstrual bleeding or endometriosis that don't impair fertility, so that women can actually, attempt to have a family whilst not suffering. Treatments in IVF, better synchronisation with embryo transfer, endometrial preparation. Treatments that don't increase obstetric risk, because there's a whole lot of data on obstetric risk increases with abnormal endometrial preparation. It goes on and on and on. If you can have an impact on the health and the lives of women, even a small impact, it's such a huge array of problems and such a huge field. that even a very small advance would make a huge difference. A blood-borne diagnostic for endometriosis. I mean, there's a Nobel Prize waiting there.
It's got your name on it, I think.
Don't think so.
This will be my last question before we open up to the floor. So if anyone has a question, be brave to put your hand up. But I guess there's a lot of students in the room, as we know. What would be your advice to them to sort of keep up with all the innovations that are coming out? You know, you guys are sort of moving with the culture of genomics, but then, you know, how do you know what to sort of grab onto, and how do you stay up to date?
I don't know, you just got to ask questions. To me, some people are naturally curious and some aren't. Maybe I dream a little bit, but I tend to see things. I go to conferences or I read a paper and you look at that and you think, wow, that you know, because as I said, the new technologies are coming all the time, and so it's not a matter of just jumping on one and that's it. You know, nowadays it changes every week, yeah, and so just keep asking questions always. Just be curious. Don't accept everything as it is, and don't prematurely turn into an ossified old person. You know, keep your mind open.
Oh, I would definitely agree with that. That was actually the first thing that came to mind for me, was curious. You know, make sure you keep curious about what you're doing, and also passionate. You know, finding an area of science that you love. That's what makes work worth coming to every day.
And sorry, there's curious and contrary. Don't believe what you're told. Don't accept what you're told. And just because someone says they're important, don't, you know, just check it for yourself, validate it, verify it, and be curious.
I want to know some examples that you got, Richard, but that's a conversation for another day.
Sorry? What?
I'd love to know an example that you're referring to, because you're obviously thinking about something, but let's not go into that. Lucky last.
Never believe the professor.
Yeah, that's true.
Yeah, I think trying to find out what's cutting edge, there's so much information out there. I get lost in the literature because there's so much coming out all the time. A lot of what's published now, I guess through peer review, is actually been out there for a while and is often presented at conferences. I mean, sometimes it can be inaccessible, I guess, for students in that regard. People like us get to go and travel to conferences and see what the big people in the field are going to publish next. I guess social media is a very good medium, although I think X has taken a bit of or Twitter's taken a bit of a dive recently. Not sure how long that will survive, but there's a really good I guess, news site called Genome Web that I subscribe to, and it comes up in my emails.
So do I, every day.
And you will see a lot of Illumina announcements. There was one today, actually, that's a bit of a win.
Not talking about that today.
So, yeah, so that's free, and you can access that. You know,
That's actually a tip that someone told me as well a few years ago, and I've done it for the last few years, but I've signed up to certain keywords that I wanted to understand. Precision medicine was one, genomics was one. So yeah, that advice someone gave me.
Search engines for your literature, like I use one called Pub Crawler. You just put your search terms and it just basically extracts everything that's coming out.
Yep.
And so, look, these are general things for searching the literature. Genome Web I find is a pretty good one for, hearing about new companies and what they're announcing as well. So.
I think Pub Crawler is such a great name. Just side note. That's my advice.
That's your advice.
That's my advice.
Sorry, we did always go to you. We've got a good 10 minutes, which is a lot of time for anyone on the floor who has any questions. Don't be shy. You don't want me to pick on you. Oh, Natalie goes first. I might give you... Do we have a spare mic, actually? Otherwise I can run around. I'll run around. No, you have to answer the questions.
Thanks. I'm really interested in your work with consumer representatives and what you see as the benefits of working with them, and on the other side, what they tell you the benefits of working with you are.
Yeah, I can maybe start with that one. So, as I mentioned, we're very involved. I think a bit of my personal background. So my older brother was blind. So I think for me, like I sort of had that right from the word go. I was very, lived experience was most important. It's really fascinating for us because advocacy and the consumer groups think that they get more out of it, and we think we get more out of it. So it's a bit of a competition. But yeah, very much all of our research projects and ideas come from discussions that we have with those people. And then with the vice versa is, they've all got us kind of on call now. So I quite often will get phone calls from people just saying, we've seen this in the media, is this actually a treatment that would be good for Australian patients, and what should we tell our community? So I love that. I love being able to sort of break down a bit of the barriers between the university and the institutes and the people that actually benefit from these. And I think another huge benefit is having those people with these conditions involved with our research team. It just keeps everyone motivated. We're very spoiled. We get to treat, young kids with these eye diseases and watch them get vision back. And that is enough, I think, to sort of keep you reminded of what you're doing.
Yeah, we've got a dedicated consumer group for cancer for non-primary, and they've been really important. I mean, I'm a clinician, I'm a research scientist, and so I don't have a lot of interface, I guess, with patients. So having that interaction with consumers is really important. So also part of an alliance, it's called the World Cancer for Non-Primary Alliance, and consumers are becoming more and more important and becoming part of meetings and conferences. So I'm going to a conference next week, called ESMO, European Society of Medical Oncology. And there's a whole session that's been organised by the consumer group, the World Cancer for Non-Primary Alliance. So this is really an essential part, I think, now of doing research, is having that interaction,
Yeah, no, consumer input drives the research agenda. It's essential. You need a partnership, but without the consumer input, it doesn't work.
Yep, I have a question. So with genomics being a port or essential to all your respective projects, I'm just wondering, what is your thoughts currently on multiomics and its impact, if you're using it now, its impact and the impact in the future of multiomics?
Oh, it's great. Absolutely the way to go. Sort of the school person level understanding. DNA produces RNA, and there's like 10 different RNAs for each DNA, and then RNA produces proteins, and there's 10 different proteins for each, and then the proteins get metabolized, and there's 10 different metabolites. So you've got each DNA, you've got 1,000 metabolites. No one system tells you how things work, and some of the most subtle changes, a protein can, the end can cleave off it, and it can turn from being a promoter to an inhibitor. So, if you really want to understand the biology of what's going on, you've got to do multiomics. And the problems start with the genes, and they end up, somewhere way into the future that we're still working on.
Yeah, multiomics is definitely the next frontier, I guess, in addition to spatial and single-cell genomics. Our project's using multiomics, and these, I guess the information we're getting from the different omics, that's been possible for some time, but now we can get this 2 different bits of information, orthogonal bits of information, from one assay, which makes it cheaper, but for clinical samples, we're often limited by the amount of material that we've got to assay in the first place, so we can get 2 bits of information rather than 1. For us, it's important to have these 2 different data types. Here we're looking at mutations and DNA methylation. It's for a diagnosis, and together they're much more powerful than each individual. I guess to understand biology, we're looking at mutations. We do a lot of work in curating mutations and trying to predict whether or not they're pathogenic, they're actually functional. If we look at RNA, we can see at least that mutation is expressed in the sample, and sometimes there's also signatures we can look at to validate that, their pathway is activated downstream of that mutation. So, multiomics is great, and then technologies. This is the hot thing at the moment around new assays for multiomics. Yeah.
And I was just going to say that even then, things like genome-wide association studies show that 80% of significant SNPs are in non-transcribed regions. So just knowing what turns into RNA and protein doesn't help you. We have so much to learn. You've got to be really humble with this. It's the answers. If the answers are easy, we'd have them.
Yep. Thanks for coming to talk today. Just want to piggyback off that last multiomics question. Do you see the cost of applying multiomics in the future going down the same way that genomics has gone down? Like, do you think it'll reach a point where everyone can have a multiomics test rather than just a genomics-based test?
Yeah, I think we're almost there actually. It's typically a bit more expensive to run an assay that will do mutation detection and DNA methylation simultaneously, but the cost is not double. I mean, that's part of the beauty of the multi-omics assays, I guess. We're getting multiple bits of information and perhaps one for free effectively. So I think that, there's different multi-omic tests or assays that are out there. I guess the one we're looking at, as I mentioned, is mutation and methylation, and we essentially make a DNA library and then we sequence that through the Illumina platform, and the result is we can detect both of these things simultaneously. So we're kind of already there, and the affordability is really in just the sequencing. As that cost comes down, I think that will be employed more. Yeah.
That's probably a good point to touch on, that all these multi-omics, when you talk about transcriptomics, proteomics, methylation or epigenomics, they all have an output which is sequencing, and most of that is moving to that space. So it's almost utilising one sequencer in multiple different ways. So I think you both picked up the good points on tonight about omics. The very last question I have, which I'm thinking about when I was a student, when I was sitting in panels, is, all right, so what job should I do now, considering all this cool stuff? Specifically in genomics, do you find there is, I guess, an unmet need in terms of skill set? Is it curation? Is it AI? Is there something that's jumped out of you that you're like, I really need this skill set, and I don't have it, or my team doesn't have it, and maybe that's the next wave of jobs out there?
I was going to say, we'll take them all for ocular genomics. So it's very much a growing space for us. So yeah, we're very much pinching people from cancer at the moment in terms of trying to find people with skill sets. It is really interesting because we're very much an emerging field. But I think one of the things that I love in this space is that people tend to move between a little bit as well. Like there's some shared skills, and that's been really wonderful to watch as well. But yeah, huge growth for people with that expertise in bioinformatics and genetic counsellors, geneticists. There's this whole area is going to grow a lot, I think.
It's a really big team, and when I look at who I've got involved, we've got everything from the consultant clinicians, the trainee clinicians, the theatre staff, because we're collecting samples from operations that are going on every day. You've got the booking clerks, you've got the people who run the medical record system, you've got the researchers in the lab extracting RNA, you've got the program manager, you've got financial people, you've got the bioinformaticians, and then you've got scientists and researchers. So there's jobs for everybody, if you know what I mean. And I think all of those people then engage and buy into the overall goal of the program. So everyone's excited about the successes, whether or not, regardless of what they actually did. Even the jaded clinicians.
Final comments, Richard?
Yeah, I think the big change I've seen in a laboratory, from the laboratory side, is that if you go into a research facility now, you'll see more people sitting at their desks on the computer than you will in the lab. Doesn't take actually that long to generate the data, and then you get this absolute tsunami of data to deal with. So any students that come through my lab now, I try to train them up in bioinformatics as well as doing the molecular biology. And I mean, this goes, I've got clinician scientists working in my group doing PhDs. They're learning bioinformatics skills. The bioinformatics is really something that's essential for genomics in particular. But I think genomic technologies are being applied across a whole swathe of different research areas, and you'll find more and more people training in this space. So my one piece of advice is, learn some bioinformatics.
Awesome. And I guess you spoke about social media. It sounds like everyone needs to go and follow them on whatever platform you guys have, which I think would be a good idea. Thank you, everyone. I'm exactly on time, and I like to be punctual. So thank you very much, and thank you to our panellists.